Okay, let's get on to the next section. So we are still in the part where we have broken apart a PIP3 molecule, or rather, phospholipase C has broken apart a phosphatidylinositol molecule, and we are following the fate of the two sections. So we just talked about what happens to the phosphorylated section, the top of the lipid, if you like. And now let's talk a little more about what happens to the bottom, which is the diacyl glycerol, DAG. All right, let's go. So we're going to run into a couple of these things, which are actually proteins that you run into a lot when you are talking about cell biology. MAP kinases are just all over the place. So those RAFs, those MEC, yeah, these are very widespread, important kinases that amplify signals and eventually lead to actually a variety of responses. All right, so MAP kinases, mitogen activated protein kinases, great. They are kinases and they're going to put phosphates on other things. RAF is going to be a serine threonine kinase, fine. And yeah, it's in this same signaling pathway as is MEC1, which is a MAP kinase, MAPKK. And yeah, it's going to be involved in the activation of the transcription factor AP1, which is actually made of two little proteins called FOS and Jun. So there's AP1 there. And yeah, actually, the ones you find in cells are called CFOS and CJun because they come from cells. Cellular version of FOS, cellular version of JUN. All right. The original genes, I think, were discovered in a herpes virus in chickens, if I remember right. And so those are the original FOS and Jun. But that doesn't really matter here, does it? Okay, ERK is another one of these kinases, extracellular signal-related kinase. And KSR is a kinase suppressor of RAS. And so that's going to be a scaffold protein that's going to be involved in this same pathway. So let's put these weird puzzle pieces together, a lot of kinases in there, and build it into a nice little whole. Oh yeah, and then you've got your Jun kinases, your junk proteins, which are going to phosphorylate cellular Jun, so they combine to cellular phos and form AP1, the very important transcription factor that's heavily involved in inflammatory pathways and cell activation. All right, how's it work? Well, just like this. So, one step before this slide, you've got phospholipase C gamma, which is cutting the phosphatidylinositol molecule, and you're going to get the two halves. The one half is going to go off and signal to the ER and let that calcium out. The other half is the diacylglycerol, which is going to bind to RAS right over here. Okay. And so this is the RAS GTP release factor, and here's our actual RAS protein. So if this is inactive RAS, and it is one of these G proteins, can you think what kind of molecule inactive RAS is holding? It's going to be a GDP, right? Because that's what G proteins do. They hold GTP for a while, then they break off the last phosphate and inactivate themselves. And once they're inactivated, they cannot be reactivated without help from a protein like one of these guanine nucleotide exchange factors. RAS-GRP is one of those. And so here we got our inactive RAS holding on to GDP, which is not pictured but is totally there. And we've got this protein that could totally bind to the back of it and open it up. But it's not going to end up in the right place unless it binds to diacylglycerol. And so once it binds, it's now going to be stuck right next to the membrane, which is right where our G protein RAS is going to be stuck. And so boom, it can make the connection and it can open up RAS. And so now RAS can accept a GTP. It will be activated. And then we are going to have a cascade. So RAS phosphorylates RAF. RAF phosphorylates MEK. MEK phosphorylates ERK, and activated ERK is going to go into the nucleus and, transcribe various things. RAS to RAF, RAF to MEK, MEK to ERK. And RAS doesn't get activated without the RAS GRP. Yeah. And the RAS GRP isn't able to activate it without diacylglycerol, which doesn't exist unless you've had phospholipase C activity. And phospholipase C is going to be anchored, of course, as you probably remember, to a PIP3 molecule, and is going to activate it in that complex. So we're going to have SLP76. Let's go back to the picture so you can see it. There we go. SLP76, we're going to have GADs in the middle, and we're going to have LAT on the edge. And that's where PLC gamma fits in, and it's the one that actually cuts the phosphatidylinositol molecule so that we can get all this wonderful signaling downstream. Okay. How does that work? Well, your ERK is going to get phosphorylated at the end of that pathway. So it's going to get phosphorylated by MEK, which was phosphorylated by RAF, which was phosphorylated by RAS. So RAS, RAF, MEK, ERK. Yeah. And then, it's going to then go into the nucleus, phosphorylate a transcription factor that it just made. And that will then transcribe the FOS gene. And once you've got the FOS gene, then the FOS can bind to a C-Jun, which has been activated by Janus kinase, I believe. Jun kinase, JNK. And together, those are AP1, and those will then change the transcriptional program. So this is just a typical cell signaling cascade. And for every signal that a cell receives, there's going to be some sort of cascade like this, because that's the point of these signaling molecules, is so that a signal from outside, in this case something that the T cell receptor bound to, can lead to a difference in transcription on the inside, which is what you see here. So that's kind of the explanation for how this all works. Okay, IKK does have that gamma subunit, which is also called NEMO, and that's fine. And remember that IKK's deal is that it's going to phosphorylate I kappa B, which will cause I kappa B to disintegrate basically over time, which will allow NF kappa B to go to the nucleus and signal. And a NEMO deficiency in basically NF kappa B signaling. You're unable to activate NF kappa B because you can't get rid of the I kappa B, is going to be this particular disease right here. Yeah. All right. So, next step in the signaling cascade. All right. So here we've got protein kinase C theta right over here, and it's going to get to the right position by binding to that diacylglycerol. So this is another protein that is going to anchor down on the diacylglycerol. And so it can only find an anchor if we've been cleaving phosphatidylinositol with PLC gamma. All right, and so once we have this, let's see, we are going to phosphorylate karma 1. And so there it is. PKC is going to phosphorylate karma 1, and so great, it has a phosphate now. And the karma 1s are going to stack together, and they're going to form a little scaffold with these proteins BCL10 and MALT1. That will then allow TRAF6 to come in, and you may remember TRAF6 from before. This is one of the proteins that was involved in some of that, what do they call them? Toll-like receptor signaling, I believe. From there, let's see, TRAF6 is going to be a ubiquitin ligase, so it's going to make a polyubiquitin scaffold. And this is not going to be lysine 48 ubiquitin. It's probably going to be lysine 63 ubiquitin. but we'd have to look it up. But just, just in case you're wondering why this doesn't lead to immediate destruction of the TRAF6 protein, it's because the ubiquitin is hooked up in a different way. So we're going to make this, which is going to bind to tab 1 and 2, which are attached to TAC1. TAC1 is then going to activate IKK. And IKK, once it's phosphorylated, is going to phosphorylate I kappa B. And I kappa B, once it's phosphorylated, is going to get polyubiquitinated and destroyed. And this is going to be lysine 48 ubiquitin, which says, recycle this protein. So it's going to get degraded, shredded in the proteasome, essentially. And that allows NF kappa B to actually go into the nucleus, finally, at last, where it can actually do transcription. So, yeah. The new parts here are just that protein kinase C theta is going to activate KARMA1. KARMA1 is going to form a scaffold with these two other proteins, BCL10 and MALT1. And then from there, we're going to recruit, TAC1 is the important part of this complex, but it comes with a tab 1 and 2. And then TAC1 is going to activate IKK. IKK is going to activate I kappa B, which is going to disintegrate, which is going to allow NF kappa B to go to the nucleus and signal. There we go. All right. So that was that's probably good for that part. We've got more. You bet we've got more. But let's break this up into bite-sized pieces so you can go back, make sure your notes are good, maybe rewatch a section. This is a chapter where you'll really benefit from rereading, rewatching. Look at the videos at the end of the chapter. All these things can help.